Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, BBRF's President and CEO. Today, Dr. Nikhil Urs will present how the brain's dopamine circuitry helps regulate cognitive flexibility and reward seeking. BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. The high quality of the research we fund is made possible by the BBRF Scientific Council. This group of over 180 prominent mental health researchers reviews each grant application and selects the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that research funded by BBRF has had such great impact is because we do not limit our focus to one illness or condition. Since BBRF, the foundation, since 1987, the foundation has awarded more than $440 million to fund more than 6,400 research grants around the world and across a broad spectrum of brain illnesses, including addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, PTSD, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Nikhil Urs. Dr. Urs is an assistant professor in the Department of Pharmacology and Therapeutics at the University of Florida. He was a 2018 Young Investigator and a 2013 Maltz Prize winner. Our webinar will begin with his presentation, which will then be followed by Q&A. To submit your questions, please use the Questions tab on the control panel on your screen. Feel free to submit your questions at any time. Following the presentation, I'll ask as many questions as possible in the time allotted. And now I'm pleased to introduce Dr. Urs. Nikhil, the floor is yours.
Thanks, Jeff. Thank you for the very kind introduction. I hope you guys can see my screen.
Yes.
Okay, thank you.
Yes.
And thank you for BBRF and NARSAD for supporting scientists like us, especially when we are in the early phase of our careers. And so I would like to get started. So the title of today's talk is Role of the Brain's Dopamine Circuitry in Regulating Cognitive Flexibility and Reward Seeking. So when we talk about dopamine, right, the first thing that comes to our mind is a reward or pleasure, right? But then we also know that dopamine plays a very important role in movement and cognition, right? So these are sort of the normal functions that we think about dopamine. But then when we think about movement, when dopamine neurotransmission goes wrong, we get disorders like Parkinson's disease, right? And then when we think about cognition, when dopamine neurotransmission goes awry in the cognitive domain, you can get disorders like schizophrenia or ADHD. Okay, and then last, the most sort of popular notion about dopamine is the sort of reward pleasure pathway, and we know like a lot of drugs of addiction like cocaine, amphetamines, they all sort of mess up the dopamine system in the brain. Okay, but what does dopamine actually do, right? So current evidence suggests that dopamine plays a major role in regulating motivated behavior. And so when we talk about motivated behavior, I just want everyone to sort of give you a very simple representation of what motivated behavior is. So usually it starts with a stimulus, maybe it's hunger, right? And then when you're hungry, you perform an action. It could be you move towards a food source, or you perform an action, or you make a choice, right? And then you get that reward or food, right? And then what happens is if you like that food, then that reward or food reward sort of reinforces that choice you made or the action that you perform. Like this puppy here that has been trained to like ring the bell, right, to get a food reward, and it keeps doing it because it's hungry, right? So when we represent this very simple sort of diagram on motivated behavior in psychological terms, right, uh, we come up with this flow chart of stimulus, response, and outcome. Okay, so you have a stimulus, right, and then you respond, you perform an action or you make a choice, and then that gets you a particular outcome. Okay, and this is classically known as instrumental conditioning or operant conditioning. Okay, and it's called operant or instrumental conditioning because the response is voluntary. You have to make a choice or you have to select a particular action to get a particular outcome. Okay, and this is also called as the goal-directed system because these are inferred responses. Okay, this is in contrast to classical conditioning or Pavlovian conditioning, where you present a stimulus and the response is involuntary, or it's an innate response that is related to the outcome. Okay, so in terms of the goal-directed system, you perform an action or you have a response and you get an outcome, and then that outcome sort of reinforces your choice, right? And so you keep doing it again and again because you know that this response will give you this particular outcome. And then eventually what happens is that the stimulus and the response become predictive of the outcome. So eventually when you perform this response to get that outcome multiple times, you eventually shift into a habitual sort of response from a goal-directed response. Okay, so what happens when you switch to a habitual response is that you have a stimulus and you respond, and you don't care about the outcome anymore because the stimulus and the response are predictive of the outcome, and so you just automatically do it thinking that, okay, if I have the stimulus and if I respond, I will get this outcome no matter what, right? And so that's the way of for the brain to sort of optimize functioning of the brain and use less energy to ensure the survival of the organism. Okay, and dopamine plays a central role in all of these systems because there has been data suggesting, especially from like rodents and primate research, that if you disrupt dopamine neurotransmission, you can affect the goal-directed system, right? But you can also affect classical Pavlovian conditioning responses as well. In addition to that, it's been shown that dopamine can also sort of affect the habitual responses, right? So dopamine basically plays a very important role in the stimulus response outcome flowchart, okay? There's another important aspect to this stimulus response outcome, and that is called cognitive flexibility, because in real life, usually what happens is that you have a stimulus and you make an action selection or you make a choice and you get a food reward. But what happens is sometimes the food reward or food source is not available anymore, right? So you have to search for a new source for food. And then you sort of have to switch from this habitual learned response and form a new association, right? And so now the stimulus and response was predictive of a particular reward that doesn't exist anymore. And so you have to sort of get out of that habit and form a new association with a new source, right? And then now you switch back to goal-directed behavior, right? And then that new food source or reward reinforces your new choice, right? Or new action selection. And then eventually, then that new stimulus action becomes predictive of this new food reward, right? And so this is basically how cognitive flexibility works. So what we are interested in is like my lab is primarily interested in is what are the neuronal circuits that regulate dopamine and how dopamine also regulates these circuits. So just to briefly give you an overview of how dopamine neurotransmission works, right? So dopamine is made from the amino acid L-tyrosine, and it's converted to L-dopa. And a lot of you might have heard of L-dopa because it's the precursor to dopamine, and it is used as a therapeutic in Parkinson's disease to replace loss of dopamine, okay? And what is also interesting is that dopamine can then be further metabolized by the enzyme dopamine beta hydroxylase into norepinephrine and then further into epinephrine. Okay, and so this is kind of important for later on in the talk. Okay, So where do we find dopamine neurons in the brain? So usually dopamine neurons are found in the midbrain, and they project to two major areas. Okay, that for the sake of this talk, I'll just focus on these two areas. So the substantia nigra dopamine neurons project to an area what is called as the striatum. Okay, whereas the ventral tegmental area, or VTA dopamine neurons, project to the nucleus accumbens and the cortex. Okay, and like I mentioned before, the dopamine is involved in cognitive flexibility, and when dopamine neurotransmission goes awry, it can give rise to multiple disorders like schizophrenia, Parkinson's, ADHD, and drug abuse. And so what is known is that dopamine, the actions of dopamine in this region called the striatum is important for movement control, whereas the actions of dopamine in the cortical regions is important for cognitive function. Okay. Okay, so coming to dopamine neurotransmission, so once dopamine, so as you can see with the green lines, it's just a cartoon showing that the dopamine neurons are projecting to various brain regions. So the first brain region is the striatum. So when dopamine, these neurons project to this brain region, and when dopamine is released, when you zoom into this striatum area, on the right here, you can see it's a presynaptic neuron. The dopamine is synthesized, it's packaged into vesicles, and it's released into the extracellular space or the synapse. This dopamine then goes on to bind to receptors on the postsynaptic side. And the postsynaptic receptors for dopamine are primarily comprised of the D1 and D2 class of G protein coupled receptors. And the D2 class of receptors are very well known because they are the primary target for most antipsychotic drugs that are used for schizophrenia therapy. So in addition to this dopamine binding to these receptors, there is a very important protein that regulates dopamine neurotransmission, and that protein is called the dopamine transporter. Okay, this dopamine transporter is responsible for reuptaking dopamine from the synapse back into the neuron. So you can think of the dopamine transporter as a vacuum that sucks back all of the dopamine back into the neuron. And so this is a very important regulatory protein that maintains dopamine homeostasis. So, and what is really important is that a lot of drugs of abuse like cocaine and amphetamines, the primary target of these drugs is the dopamine transporter. So they disrupt dopamine transporter function, and that's why you have elevated dopamine levels in the extracellular space or the synapse when these drugs are administered. So that is in the striatum, where you have a lot of dopamine transporter. What is interesting is that even though you have dopamine projections to the cortex, for example, in the PFC, there is little to no dopamine transporter expression in the PFC. However, there are these norepinephrine neurons that arise in the locus ceruleus in the hindbrain that also project to the cortical areas where the dopamine neurons are project to. And these norepinephrine neurons express their own transporter called the norepinephrine transporter, and they reuptake norepinephrine. And if you remember from a couple of slides ago that norepinephrine is basically metabolized from dopamine, so they're very closely related. And so what happens in the PFC is that the norepinephrine transporter reuptakes dopamine and basically functions as the transporter for dopamine. And so this is a very interesting sort of distinct regulation of dopamine in the striatum versus the cortex. So in the striatum, the dopamine transporter maintains dopamine homeostasis, whereas in the cortex, it is the norepinephrine transporter that maintains dopamine levels. Okay. So in addition to this sort of distinct regulation of cortical and striatal dopamine, there have been several studies that have shown that there are opposing levels of cortical and striatal dopamine. And what I mean by that is, like, for example, when you administer rodents with a low dose of methylphenidate, and methylphenidate is basically the generic name for Ritalin, which is an ADHD therapeutic. And when you administer rodents low doses of methylphenidate, you see in these black squares here, this is dopamine in the cortex. And the dopamine levels in the cortex are much higher compared to the dopamine levels in the striatum shown by these black circles. However, if you, what I don't show here is if you inject a high dose of methylphenidate, you see the exact opposite. Now the striatal dopamine, or the black circles, are much higher, and the cortical dopamine is, the cortical dopamine is much lower, again suggesting that there is this opposing levels of cortical and striatal dopamine. Furthermore, some groups have found that there are certain strains of mice, here the C57 and the DBA strains, and what they saw was that the DBA strain, which is in the dark triangles here, they have elevated cortical dopamine, but they have reduced striatal dopamine. Okay. However, the C57 strain of mice, which are in the dark circles here, they have low levels of cortical dopamine but higher levels of striatal dopamine, again suggesting that there is this opposing levels of cortical and striatal dopamine. So whenever there's high cortical dopamine, there's low striatal dopamine, and vice versa. So the last piece of evidence sort of solidifying this observation are these mice called the NET knockout mice. So if you remember, I told you before that the NET, or the norepinephrine transporter, is the primary mechanism of clearance of dopamine or reuptake of dopamine in the PFC. Okay, so if you make mutant mice that lack the gene for this norepinephrine transporter, you lose that reuptake function, and you elevate both norepinephrine and dopamine levels in the cortex. Okay, surprisingly, what was observed was that in this mice, when you see this elevated dopamine and norepinephrine in the PFC, you see a parallel reduction in striatal dopamine levels, again suggesting that there is this sort of opposing regulation going on between the cortical dopamine and striatal dopamine. So what we wanted to test was, the first thing was that, can we somehow pharmacologically manipulate the norepinephrine transporter in the PFC and modulate striatal dopamine? So we turned our attention to a model of striatal hyperdopaminergia. And you remember I told you that in the striatum, the DAT is what clears dopamine, and if you inhibit the function of DAT, you get elevated striatal dopamine levels, right? And so if you make a mouse model, similar genetic model, where you knock out the gene for the dopamine transporter, you get elevated striatal dopamine levels. And here is a graph showing the hyperactivity of these mice. So these dark circles here are the DAT knockout mice, and their control wild type litter mates are here down below. As you can clearly see that the DAT knockout mice are very hyperactive, and these were at that time considered to be a very good model for the hyperactivity seen in ADHD, because when you administer drugs like methylphenidate or amphetamine, which is basically Ritalin and Adderall, you see this calming effect of these stimulant medications, which was at that time called as the paradoxical calming effect of these stimulant medications. And so we have this very nice mouse model where you have striatal hyperdopaminergia, and we wanted to test, okay, if we inhibit NET or norepinephrine transporter function specifically in the PFC, can we reverse the striatal hyperdopaminergic behavior? Okay, so the first thing we did was we first tried a systemic injection. So we just injected these mice IP, so it goes all over the body, and we used two specific drugs. So both methylphenidate and amphetamine are very non-specific drugs. They can bind to multiple transporters, and they can elevate levels of norepinephrine, dopamine, serotonin, and all of these monoamines. And so we tried specific transporter inhibitors called desipramine and fluoxetine. And desipramine is a specific inhibitor for the NET, whereas fluoxetine, or Prozac, which is the trade name, is a blocker of SERT, or it's an SSRI. And so when we systemically inject these DAT knockout mice with either desipramine or fluoxetine, we again see this inhibition of the striatal hyperdopaminergia-induced hyperactivity. And so we just repeated what was observed before, but now the next question was, can we inject these drugs directly into the PFC? And so what we did was we did a direct PFC infusion of both desipramine and fluoxetine into the PFC of these DAT knockout mice. And as you can see in the vehicle or control mice, the DAT knockouts remain hyperactive, but when you inject or infuse desipramine into the PFC, we see this inhibition of the hyperactivity. However, surprisingly, we did not see an inhibition of the hyperactivity with fluoxetine, suggesting that maybe fluoxetine does not work through the PFC, but through some other mechanisms. And so we were really excited by this finding that, okay, we can actually manipulate net function in the PFC specifically, and we can increase dopamine and norepinephrine levels in this region that can sort of reverse the striatal dopamine-induced hyperactivity. But as you can see in this cartoon, that inhibition of NET increases both dopamine and norepinephrine. So we wanted to ask the question, okay, is it actually dopamine or is it norepinephrine? And to get at this, we used a drug called as nepicastat. So nepicastat is, so highlighted here down here in red, is an inhibitor of the dopamine beta hydroxylase enzyme that converts dopamine to norepinephrine. And so if you administer nepicastat to mice, you block the conversion of dopamine to norepinephrine and you accumulate dopamine. So you effectively elevate dopamine levels, but you deplete norepinephrine levels. And what was really exciting to us was that when we did either a systemic or a PFC infusion of nepicastat, we saw an inhibition of this hyperactivity in the DAT knockout mice. Again, suggesting that it is not norepinephrine, but it's actually dopamine, that is dopamine levels in the PFC that is reversing this striatal hyperdopaminergia and hyperactivity. So just to summarize quickly this part, we showed that PFC modulation of dopamine levels can reverse the hyperactivity in the DAT knockout mice, and that this is specifically due to an elevation in dopamine levels and not in norepinephrine levels. However, coming back to the very interesting finding that fluoxetine did not work when we infused it into the PFC. Some studies have shown that systemic injection of fluoxetine can actually elevate dopamine, but that's through a slightly different pathway. We know that the dorsal raphe neurons, which are the serotonin neurons, project to these PFC dopamine neuron cell bodies in the midbrain and can regulate their activity. And so fluoxetine might be working through an indirect mechanism where it regulates the activity of the dopamine neurons that project to the PFC and then cause release of dopamine. But nevertheless, what was the common finding was that in both cases, when you inhibit NET function or you deplete norepinephrine function, in both cases, you're elevating dopamine. And then this elevated PFC dopamine may represent a common mechanism for ADHD therapy. So just switching gears now, so we sort of looked at how cortical dopamine can regulate striatal dopamine-induced hyperactivity, but we were very much interested in how these cortical striatal dopamine circuits are influencing cognitive flexibility and this reinforcement learning, right? So remember, you have a stimulus, and then you perform an action or a choice, and you get a food reward. When that reward is not available anymore, you have to find a new source and sort of form a new association, and then that new reward sort of reinforces your choice, right? And so this is called as reinforcement learning, because your brain learns that this reward is beneficial, it's good for survival of the organism, and so I should keep making that action selection or choice, and eventually that stimulus and response becomes predictive of that outcome or reward. Okay, so to test this cognitive flexibility, we use this task called as the probabilistic reversal learning task. So it's a very simple task where we train the rodent, a mouse in this case, we train it to nose poke or press a lever that has a high probability of getting a reward, and the other lever or nose poke has a low probability of getting a reward. So the mouse very quickly, within a week or so, learns that it needs to stick to this high probability lever or nose poke because it has the highest chance of getting a reward at this high probability nose poke compared to the low probability nose poke. And so what we do is eventually, after a few trials where the mouse sticks to it continuously to this high probability lever, we switch the contingencies. So now the previous low probability lever becomes a high probability lever now. And so now the mouse has to now learn again, oh, the previous one is not the high probability lever anymore, it's the other one. So now it has to relearn and form a new association. And it has to now stick to that for multiple trials, right? And then after it does that, then we switch it back again. And we keep doing this multiple times during an entire session. And so we measure how many times it can switch from one nose poke or one lever to the other. And that is a measure of cognitive flexibility, okay? So I won't go into sort of the details, but we train the mice over many days to sort of learn to where it needs to nose poke or lever press to get a reward, okay? And then eventually, for the last week or so, we train them or let them perform this task of probabilistic reversal learning, okay? And so we again took the net knockout mice, right? Remember, the net knockout mice have elevated dopamine and norepinephrine, but they have reduced striatal dopamine. And we can, when you manipulate this cortical striatal dopamine, you can reduce the hyperactivity. So we also asked the question, can these mice, do they have an altered performance in cognitive flexibility tasks? And sure, when we put them through this probabilistic reversal learning task, the net knockout mice significantly outperform their wild type controls. Like almost they have like double the reversals compared to the wild type controls, which was very exciting to us at the time, because not only does this mechanism like reduce the hyperactivity, but it also improves their cognitive flexibility. What was also very interesting to observe was that these net knockout mice have fewer omissions per session. So what that means is that they go through these multiple trials, and so their wild type controls, they don't perform on all trials, so they sort of take a break, right? Whereas these net knockout mice, they perform on pretty much 90% of all the trials. And so their one interpretation of that data is that they're extremely motivated to like perform this task, right? And also they have more nose pokes per session, suggesting that they're really engaged and they really want to nose poke to get the reward, okay? So in addition to this cognitive flexibility task, what we did was we came back to the same question. Okay, in the net knockout mice, there's elevated dopamine and norepinephrine. So which is it? Is it dopamine or is it norepinephrine? So we used a slightly different approach here because we wanted to sort of use the strongest possible approach. And so what we did was we lesioned the norepinephrine neurons, the terminals in the PFC. So we did this by injecting a toxin called as 6-hydroxydopamine, which is uptaken into the norepinephrine terminals, into the neurons, and it kills those neurons, right? And so you deplete norepinephrine levels. Surprisingly, what we saw was that even after depletion of norepinephrine levels, we still saw that their reversal learning was pretty much intact compared to the control mice. However, what was interesting was that even though their learning seemed intact, we saw two major changes. One was that after the lesions, right, these mice performed on fewer trials, so their omissions were higher, which is the exact opposite of the net knockout mice, right? So even though they're performing on fewer trials, they have a lot more omissions, they are still having the same number of reversals. So again, this kind of suggested that maybe norepinephrine is involved more in the arousal or the alertness part of this task, whereas dopamine is actually involved in the actual reinforcement learning or making that choice to get maximum rewards, okay? And so lastly, what we did was we also sort of wanted to look at this switch from this goal-directed behavior to this habitual behavior, right? So remember that in goal-directed behavior, you're constantly making a response or you're making a choice and you're getting a particular outcome, and then you keep repeating that choice because you're getting that outcome. So that is a goal-directed performance, right? And eventually, when the stimulus and response become predictive of this outcome, you sort of dissociate from that outcome and you just stick to the stimulus response task, right? So we can sort of mimic this stimulus response switching from a stimulus response outcome switching in mice using this goal-directed habitual task. And so I won't go into the details, but in this task, we sort of train the mice to constantly lever press to get a reward, right? So early on in training, right, you can see that these mice, the wild type mice early on in training, they are sensitive to devaluation. So what we do is like before the test happens, like 1 hour before, we satiate them. So we give them the reward, and so they're satiated, so they should not respond anymore, right? And so we see that, yes, we see a reduction in their responding, right, or lever pressing. The same thing with the net knockouts early on in training. You know, if we satiate them, you know, their motivational drive is reduced, and so they respond less. However, when you keep overtraining them, right, eventually they switch to this habitual response where they are now dissociated from the outcome. So what happens is, even though you satiate them, their responding doesn't change. It is not significantly different from their non-devalued controls, right? However, in the net knockout mice, you still see a significant reduction in their responding, suggesting that these mice are still very much goal-directed and showing goal-oriented performance. So again, all of these data were suggesting that these net knockout mice are not only more motivated to perform a task, but they are more goal-directed, okay? And then that dopamine is central to all of this because, as I showed you before, that in the net knockout mice, we have elevated cortical dopamine, but reduced striatal dopamine. So, okay, well and fine. Okay, you know, we had this really interesting observation. Okay, the net knockout mice, when you manipulate norepinephrine transporter in the PFC, you reduce hyperactivity, but also if you manipulate cortical dopamine levels and reduce striatal dopamine levels, they perform. They are more cognitively flexible. They are more goal-oriented, right? But how is this exactly happening? Like, what is it doing to dopamine? And is there actually a change in dopamine that is causing this enhanced performance, right? So to test this, we wanted to measure striatal dopamine dynamics. And so one of the very well-known sort of phenomenon in the dopamine field is that dopamine neurons have sort of these two types of firing modes, right? So there is a tonic sort of baseline neurotransmission that is a spontaneous firing of the dopamine neurons that lead to very low baseline levels of dopamine. And then there's the phasic transmission or phasic burst of dopamine, which is caused by actual action potentials, which leads to this phasic burst of dopamine, and very large amounts of dopamine are being released that activate various dopamine receptors, and then that can be cleared by the dopamine transporter. And so what is thought is that, or has been also shown, that these phasic bursts of dopamine are related to various events that are encountered by an animal. So if an animal moves, you see a burst in phasic dopamine, right? Or if the animal gets a reward, you again see a burst in dopamine. But how do you measure these tonic or phasic bursts of dopamine in a live mouse or an animal that is performing this cognitive flexibility task? And so we turned our attention to this technique called fiber photometry, where they created this really cool tool called D-Light, which is a fluorescent dopamine sensor. So whenever dopamine levels go up, the fluorescence increases. When dopamine levels go down, the fluorescence decreases, okay? And you can measure changes in dopamine at the millisecond level. As you can see here, there's a great example of how this sensor, when you get a foot shock, which is an aversive stimuli, you see like this downward a negative response in dopamine. But if you get a reward, you would see a positive peak in dopamine. Okay, so we use this fiber photometry technique in our wild type and net knockout mice to see that are there any differences in tonic versus phasic burst in the wild type controls versus the net knockout mice, which have sort of enhanced performance in this cognitive flexibility task. And so we did some pilot studies, and as you can see in this figure here, this gray line represents when the pellet or reward was administered or was given to the mouse, right? And you immediately see this very nice positive dopamine peak that goes away. So this is a phasic burst in dopamine, right? So, and as expected, on the right side here, when the pellet was not given to the mouse, so the mouse presses a lever or nose pokes for the reward, but it doesn't get it. So you immediately see this negative dopamine response, right? So you can see that whenever, and this is how sort of reinforcement works, right? So when you get a reward, you see a positive dopamine response, and that reinforces and helps the brain learn that, okay, if I perform this action, I get I get a reward. But if I perform this action and I don't get a reward, then dopamine goes down, and then you associate that choice less with a particular outcome. Okay. So what happens when we use this dopamine sensor in our wild type and net knockout mice? So remember, the very first thing that we talked about in motivated behavior was the cue or the stimulus, right? So in this case, the stimulus is the light turning on in the chamber where the mice are, right? So as soon as the light turns on, you can see that there's this huge spike in dopamine in the wild type controls. And what you can appreciate is that in the net knockout mice, this stimulus or cue induced dopamine is twice the size of the peak in the wild type mice. So if you look at the y-axis, this is close to around 8, whereas here it's sort of between 3 and 4. And we have seen this over multiple days. So we're very excited by these results because we can actually see that in the net knockout mice, the stimulus induced phasic dopamine peaks are twice the size of the wild type mice. And as expected, when the mice get the actual reward, there's also a phasic burst in dopamine, which is around here 4, whereas in the net knockout mice, it's like around 10. So it's like more than twice the size of the wild type controls. And so again, this is the stimulus, and then the bottom one is here. When you respond, you make a choice at the high probability nose poke or lever, and then you get a reward, right? And so you see this really big rise in dopamine, like a phasic burst. Interestingly, when the mice make a choice at the low probability nose poke and they do get a reward, you still see a rise in dopamine. And interestingly, when at either of these nose pokes, whether it's the high probability nose poke or the low probability one, when they don't get the reward, you see this negative dopamine peak. And what was really exciting and interesting for us was that the negative peaks are also twice the size in the net knockouts compared to the wild type mice. So you can see that the negative peaks in the wild types are around -2, whereas in the net knockouts is -4 or more. So again, it was like the phasic bursts, whether they're positive or negative, they're much bigger in the net knockout mice compared to the wild type mice, again suggesting that maybe because these phasic bursts are so much more bigger in the net knockout mice, the stimuli and the responses are much more reinforcing, and that's why they are much more the association of the stimulus and response with the outcome is much more stronger, and that's why they are sticking to a more goal-oriented task like behavior. However, what was really interesting to us was that not only were these phasic bursts like really big, but we made an interesting observation that the baseline dopamine levels were also much smaller in the net knockout mice. So the blue line here is sort of the baseline dopamine level, and then as you can see, there are these spikes, which are the phasic dopamine burst, and the dark blue line is sort of the baseline dopamine. And if you compare that to this control signal here, the difference is about like 30, right? Whereas in the net knockout mice, the difference between the control and the dopamine signal is only about 10. So again, there's almost like a 50% decrease in the baseline dopamine levels, but the phasic dopamine bursts are like double the size in the net knockout mice. So again, this kind of brought us to our hypothesis is that yes, you have these two modes of signaling, the phasic and the tonic, right? And the phasic dopamine bursts are more correlated with events like presentation of stimulus, with responding, or with the outcome, which is like getting a reward, or even like, you know, avoiding an aversive stimulus. Okay, and so we hypothesized that the tonic level of striatal dopamine is what sort of regulates or allows for this increase in phasic dopamine bursts, right, which are much larger in the net knockout mice because there is an enhanced signal to noise ratio. And so just to summarize this part, you know, we showed that the net knockout mice have enhanced cognitive flexibility and goal-directed but less habitual behavior. And maybe because these net knockout mice have higher phasic responses and lower tonic baseline levels, they have enhanced motivational drive to perform this task, right? And what was really interesting to us was that even if you deplete norepinephrine, you don't affect reversal learning per se, but maybe you're reducing the motivational salience, which is maybe reflected by the arousal or the attentional state to different stimuli. Okay. And so just the last part of the presentation is, okay, so we have this really interesting observation that you have this corticostriatal regulation of dopamine, and that the cortex, the cortical levels of dopamine might be regulating the baseline striatal dopamine levels. And so when you have higher cortical dopamine levels, you have lower striatal dopamine levels. And because you have these lower striatal dopamine levels, you allow for bigger phasic bursts of dopamine that help the animal form stronger associations with various stimuli, or to the response, or even to the outcome. And because of these more stronger associations, these mice or these animals stay with a more goal-oriented task, and they're more motivationally driven to perform that task. Okay, but the big question is, okay, yes, we see this corticostriatal interplay, and we see changes in phasic dopamine and tonic dopamine, but what are the circuits that are actually mediating this effect, right? And we are still sort of in the infancy stages of trying to figure this out. But as I told you before, that, you know, dopamine is released, and both norepinephrine and dopamine are released in the PFC, right, and in the striatum. And then when this dopamine is released, they bind to dopamine receptors on the postsynaptic side, the D1 and D2 receptors. And what we know is that there's a lot of dopamine D1, D2 receptors in the PFC and also in the striatum, right? And as I told you before, that the D2 receptors are the primary target for a lot of antipsychotic drugs, but they primarily target the striatal D2 receptors, right? So we wanted to know what are the expression patterns of the D1 and D2 receptors in the PFC. And so I won't go into too much of the details, but what we published this a couple of years ago, where we showed that the D1 and D2 receptor expressing neurons in the PFC have a very distinct pattern of expression. What we saw was that the D1 neurons, right, they have this topographical expression where the more dorsal or prelimbic D1 neurons project to the dorsal striatum, whereas the more ventral or D1 receptor neurons in the orbitofrontal cortex project to the nucleus accumbens core. Okay. However, what was interesting was that none of the D2 prelimbic neurons projected to the striatum. They projected directly to the midbrain dopamine neurons. So again, this suggested to us that once dopamine is released in the PFC, it is activating both D1 and D2 receptors on these distinct D1 and D2 neuronal populations that can project to the striatum or to the midbrain dopamine neurons. And so we were super excited by these findings, because now is the question, okay, if you are increasing cortical dopamine levels and you're going to differentially activate or inhibit these D1, D2 PFC neurons, and then those neurons are going to project to different areas of the striatum and the midbrain, and they're going to regulate both dopamine release and activity of other neurons. So we sort of came up with this revised model where previously it was known that prelimbic neurons project to the more dorsal part of the striatum, whereas the infralimbic orbitofrontal neurons project to the core or the ventral part of the striatum. But now we know that even it's not as simple as just prelimbic or infralimbic, but even within that, you can sort of segregate these regions based on their dopamine receptor population. So like where the D1 prelimbic neurons project to the dorsal striatum, whereas the D2 prelimbic neurons are projecting back to the midbrain dopamine neurons and changing dopamine release. And one of our hypotheses is that these D2 dopamine neurons might be regulating the tonic baseline levels of dopamine that we measure in the striatum, and that might be leading to changes in phasic dopamine bursts in the net knockout mice that lead to this enhanced cognitive flexibility. So just to summarize everything I've shown you so far, is that we showed that the net knockout mice have this elevated cortical dopamine levels and a lower striatal dopamine level. And so now we want to sort of ask in the future is that how are these striatal dopamine levels regulated by these cortical norepinephrine and dopamine through these pyramidal projection neurons, D1, D2 expressing neurons that are primarily glutamatergic neurons that project to the striatum or to the midbrain, right? And we've shown that the net knockout mice have enhanced cognitive flexibility, but what are the individual contributions? So I should give you sort of an insight that even if we reduce norepinephrine, we can still have normal reversal learning, but we change the attentional state of these mice. And so now what we want to do is manipulate just these dopamine neurons that release dopamine in the cortex, or the norepinephrine neurons that release norepinephrine in the cortex, and see how they individually affect different aspects of dopamine dependent cognitive flexibility. The other thing that we were really interested was, okay, so we showed that there is clear evidence that these net knockout mice have this enhanced motivational drive. So how does that affect drug seeking? So we are in the process of now sort of testing out if these net knockout mice have a different ability to self-administer for cocaine and other sort of drugs of abuse. Like I said, we are very much interested in mapping out the different dopamine circuits in the PFC, the dopamine receptor circuits in the PFC, and how they regulate not only behavior, but these phasic and baseline levels of dopamine. And lastly, what is very crucial and is very close to my heart is like pharmacological manipulation of these circuits. Like, can we identify unique compounds that can manipulate these circuits? For example, we've identified nepicostat, which is a dopamine beta hydroxylase inhibitor, and we very clearly show that it can inhibit hyperactivity in that knockout mice. So can nepicostat be potentially used as a alternative therapy for ADHD or some aspects of in schizophrenia or other disorders where there's a problem with cognitive flexibility? And so we are currently trying to find such pharmacological approaches where we can manipulate these circuits. And so that's the last slide for my presentation. I just wanted to acknowledge all my lab members. So the ones with the asterisks are former lab members who did most of this work, and then Rosa and Mariana, who are current graduate students. And and we couldn't do this without all the undergraduate students who are so helpful in running all of these behavioral tasks, which are sometimes very time consuming. And my collaborator at UF, Barry Setlow, who was instrumental in helping me set up a lot of these behavioral paradigms, and Stan Floresco at UBC, who also helped us with the probabilistic reversal learning task. And I can't say thank you enough to BBRF and NARSAD, because I've had a great relationship with them for more than 10 years. Back in 2013, when my mentor, who passed away recently, Mark Caron, who won the Lieber Prize, and he nominated me and another postdoc for the Sidney Bayer Prize, which is for trainees, and also the NARSAD Young Investigator Grant, which was instrumental in doing some of the initial studies that I showed you with the hyperactivity. Could not have been done without those funds. So extremely grateful to NARSAD and BBRF. And then finally also the NIH, through whom I've been fortunate enough to get multiple funding sources to keep exploring these ideas in my lab. And so with that, I'm going to end my talk. Thank you so much for your attention. And please, if you have any questions, feel free to ask any questions you have. Thank you.
Good. Thank you, Nico. Excellent presentation. Very interesting and exciting work that has the potential to make a difference for people. And thank you for speaking about those earlier grants from BBRF. That's the goal that we have, to get new ideas and new careers going, and you're a perfect example of that. So thank you.
Yeah, thank you.
I'd like you to expand a little bit, because you touched upon it, and I think it's important. Some of that translational stuff, how you're looking to use this information to potentially come up with new treatments. And I'd like you to speak a little bit about where you see this going in terms of new treatments that may be developed as a result of this important basic research.
Right. Yeah, that's something that I think about like every day, right? I mean, because that's the ultimate goal, is to, can we make new or better therapeutics, right? And so, for example, I talked a little bit about nepicastat, how that could be one way to sort of alter dopamine levels, but then also you're altering norepinephrine levels, which might be slightly different from current medications like methylphenidate or Adderall, that non-specifically increase multiple neurotransmitters in the brain. But also we also have to sort of start thinking in terms of like neuronal circuits, right? Because to me, at the current rate at which we are going, is that all of these different what we are finding is that there are all of these neuronal circuits, and there might be, for some disorders, there might be overlapping circuits, right? But there might be unique circuits that are dysfunctional only for that particular disorder, right? So for example, when we talk about cognitive flexibility, you can see problems in cognitive flexibility in multiple disorders: ADHD, schizophrenia, OCD, right? So it's possible that there's a common circuit that manipulates or goes awry, which is responsible for cognitive flexibility. But then there's unique circuits that are unique to those disorders. Like, for example, there might be other circuits that are going awry, that are causing hallucinations and delusions in schizophrenia, that are not necessarily dysfunctional in ADHD or OCD, right? So I think that's kind of the future, where we need to sort of find out, like, what are these individual circuits, neuronal circuits doing, right? And how they overlap between various disorders. And then obviously then the next step is, how do we manipulate those circuits, right? I mean, there's newer tools like optogenetics, but those are sort of like very invasive tools. There's a transcranial magnetic stimulation, but again, that's very non-specific. And so there's we are limited by the tools that we have, right? So I think tool development is like the next sort of frontier for neuroscience, where we can sort of target these specific neuronal circuits. And one of the really interesting tools that I really I have really high hopes for and I really like is this tool called DART, D-A-R-T. Excuse me. So this was done by a scientist at Duke, Mike Tedros, and so it sort of mixes pharmacology and circuitry, so you can sort of target drugs to a particular circuit, right? And so if these such kind of tools can be further developed, right, we can specifically target drugs to a particular circuit, then we could envision, okay, can we manipulate pharmacologically this circuit that is dysfunctional in terms of cognitive flexibility, but then another drug that can sort of reverse hallucinations and delusions in schizophrenia, and so on and so forth. So that's kind of where I envision where therapeutics is going to go towards.
Thank you. Very good overview of that. I'd like you to tell us a little bit about how the studying mice then relates to understanding these issues in human beings. What are the similarities between the mouse brain and these circuits and the human brain that allows us to get so much information in studying the mice?
Right, and that's a great question that we as scientists, debate all the time, right? I mean, how do we translate rodent or even primate work into humans, right? So if you think about some of the initial slides that I showed you about motivated behavior, these are very basic behaviors. They are found in all mammals, right? Even like the smallest mammal, or even some invertebrate animals, that is like a very basic behavior, right? Will work for food, right? So if you're motivated, you will perform a task to get a reward, right? Because that is essential for the survival of that organism, right? And so these circuits are sort of like evolutionarily ingrained in all mammals, right? And so you can sort of study these basic circuits looking at cognitive flexibility, reward seeking, reinforcement learning in mice, and they really translate really well into humans, because a lot of studies, cognitive tests that are performed in humans, we try to mirror those cognitive tests in mice to the highest extent possible, right? And so obviously we can't ask questions to mice, but we try to keep them these tasks as close to all the cognitive tasks that are done in humans. So yeah, I mean, it's a very important question, but I think the fact that these behaviors are so basic evolutionarily that we can translate from mice to humans. Obviously in humans it gets a little bit more complex, because you start involving emotional aspects of behavior, and so that makes things complicated. But these basic sort of drivers of motivated behavior are conserved throughout all mammals.
Great, thank you. I want to, Nicole, again, thank you for joining us today and for all the work that you have done and continue to do. Thank you so much.
Yeah, and thank you, and thanks for giving this opportunity to share my research. And to whoever's in the audience, if you have any more questions, feel free to email me anytime.
Great, thank you for that.
Thank you.
I also want to thank everybody who joined us today. I'm pleased to share that BBRF has received a $1 million challenge match for 2023 from two very generous family foundations that are passionate about our mission. The goal of the match is to accelerate the momentum in brain research as further breakthroughs are still needed. You can participate in this initiative if you're a new BBRF donor, a past donor who has lapsed but makes a new donation this year, or a current donor who increases your 2023 gift with your increased amount to be matched. I hope you'll consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants. This means that when you donate $1 for research, that dollar goes directly to the scientist. To make a gift, please visit bbrfoundation.org or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it with family and friends, please visit the events and webinar page on our website. And join us again on June 13th at 2 p.m. Eastern Time, when Dr. Kate Fitzgerald, the Ruane Professor of Child and Adolescent Psychiatry at the Columbia University College of Physicians and Surgeons and the New York State Psychiatric Institute, will present Developing New Treatments for Childhood Anxiety and OCD. Can cognitive control help kids grow out of illness? Thank you. And remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you.